My name is Abhishek Dimre, and we have already studied our part 1 of sexual reproduction in flowering plant, and now this is our part 2 of sexual reproduction in flowering plant. In our first part we have already studied gametogenesis, or in our second part now we have to discuss gamete transfer. So in sexual reproduction we have already studied pre-fertilization event, gametogenesis, and now we have to discuss our second event known as gamete transfer. So gamete transfer is basically known as pollination. So pollination, what is mean by pollination? It is a process in which transfer of pollen grain from anther to stigma. So anther to stigma pollination is carried out or takes place. But that pollination, here in this diagrammatic representation, you can clearly observe that transfer of pollen grain takes place by A, B, C type. So that A indicates when transfer of pollen grain from anther to stigma of a same flower. B indicates transfer of pollen grain from anther to stigma of a different flower. But A and B indicates of same plant. Same plant. You can observe, see that here. But C indicates transfer of pollen grain from anther to stigma of a different flower. So different flower of a different plant. So overall, a definition which created or occurred in pollination is, it is a process in which transfer of pollen grain from anther to stigma of same flower or different flower or same plant or different plant of a same species or different species known as pollination. So this pollination basically consists two major types, and these two major types basically carried out or takes place in two different spermatophytes. So let's discuss about these types. Pollination basically divided into two types, that is direct pollination and indirect pollination. So direct pollination means transfer of pollen grain direct towards the ovule, and indirect pollination means transfer of pollen grain first to stigma and then come to style and then come towards the ovule. So direct pollination basically shows that pollen grains stick to ovule, and indirect pollination say that pollen grains stick to stigma and then it come towards the ovule. So gymnosperm basically includes direct pollination and angiosperm includes direct pollination. So we have to discuss sexual reproduction in flowering plant, or in that sexual reproduction in flowering plant we were discussing about the indirect pollination. So let's discuss about. So pollination, here a tree diagrammatic representation of pollination which indicates two major types of pollination. From that, first type is known as self-pollination and second type is known as cross-pollination, where self-pollination is again divided into two types, that is autogamy and geitonogamy, and cross-pollination is divided into again two types, that is xenogamy and hybridization. But we have to observe here agencies of pollination, and here two agencies of pollination. From that, one is non-living agency known as abiotic factors, and biotic agency, biotic factor mean living agencies. So non-living agencies are wind and water, and biotic agencies are animals, which is simply known as zoophily. So first we will discuss about the self-pollination. What is mean by self-pollination? Self-pollination means the transfer of pollen grain from anther to stigma of the same flower or another flower of the same plant. So in first diagrammatic representation you can observe, see that 1:1. It indicates transfer of pollen grain from anther to stigma of a same flower of same plant. Or in 2:1 in second diagrammatic representation, transfer of pollen grain from anther to stigma of a different flower but of a same plant. Here, two types of pollination. From that first type is known as autogamy and second type known as geitonogamy. So what is autogamy? How autogamy takes place or occurred? So autogamy, that name indicates automatically pollination takes place or occurred, and that pollination takes place directly in that flower. You can easily observe, see that pollination takes place within same flower, and that pollination takes place because of few circumstances, and these circumstances are known as chasmogamous flower, clistogamous flower, or bird pollination. So what is mean by chasmogamous? Chasmogamous means flower exposed and open to anther and stigma. What is mean by clistogamous? Clistogamous means closed flower where anther and stigma are closed because of special estivation. That estivation is known as vexillary estivation. And bird pollination, in that where flowers are close in condition, because of that in bird condition pollination actually takes place or occur. So through example we can clarify these circumstances. Here are the examples of autogamy, chasmogamous flower, clistogamous flower, and bird pollination. So examples are convolvulus, catharanthus, gardenia, mirabilis, sunflower, potato are the example of chasmogamous flower. Clistogamous flower, close flowers, commelina, balsam, viola, oxalis, groundnut. And bird pollination, wheat, tobacco, tomato, linseed, jute, rice are the bird pollination. And here the few examples of geitonogamy. Geitonogamy means neighbor pollination. So here you can observe, see that same plant, that is known as monoecious plant, but it consists two different type of flower. Unisexual flower, at upper side presence of male flower, at lower side presence of female flower. So male flower produce n number of pollen grains, and that will stick to the stigma of lowermost flower, and easily pollination takes place or occur in that same plant. Example maize, cucurbita maxima. These are the examples of geitonogamy. After autogamy, we will discuss about the second type of pollination, that is cross pollination, and it involves agents of pollination. And to perform cross pollination, we required agents like abiotic agents and biotic agents. So in previous tree diagrammatic representation we can already observe that, but here names are given with their types of pollination. So wind agency, and that type is known as anemophily. It is also known as commonly wind pollination. Agency is water. That type of pollination is hydrophily. So most commonly in NEET examination, these questionnaires were asked. Insect pollination is also known as entamophily. Bird pollination is known as ornithophily. Bat pollination is known as chireptophily. Snail pollination is known as malcophily. Like these questionnaires were most commonly asked in NEET examination. So let's discuss our first abiotic agencies of pollination, and it includes first type known as anemophily. So anemophily, what that indicates? Anemo means here wind pollination. Pollination takes place with the help of wind. We can clearly observe, see that with the help of wind pollination is carried out or takes place. So because of few circumstances, because of few adaptation, that flower basically show adaptations. So that adaptation includes floral adaptation, and because of that floral adaptation, that plant is known as anemophilous plant. So anemophilous plant consists of different, here we can observe, see that different type of floral adaptation. So what are the floral adaptation which actually carried out or takes place in anemophily, that we have to discuss or observe here. So in anemophily, first flower, we can observe, see that flower is hidden in nature, that is known as inconspicuous in nature. Flowers are small, so flowers are non-colored. It means you can observe, see that white color flowers most commonly present in anemophily. That flower is showing here large number of pollen grains. You can observe, see that here large number of pollen grains which can liberate, because loss may be created or occurred. So because of that, many number of pollen grains will produce or occur, like camelina, maize or tassel of maize produce 25 million pollen grains. So lots of number of pollen grains were produced or occurred in anemophily. Anther is having a particular type of shapes, that is known as versatile anther. Versatile type of anther is there. That pollen grain is lightweight and non-sticky. So non-sticky pollen grains are there. Lightweight pollen grains are there. Pollen grains sometimes having a special wing shape structure known as pollinia. So pollinia is also present or occurred in that. No nectary glands were produced or bright colors were produced or occurred in that. So here are the few agencies of pollination which actually occurred or takes place in anemophily. Second type, that is known as hydrophily. Hydrophily, that word indicates water. Agency is water. So in most of the monocots, actually this hydrophily is carried out or takes place. Common examples are there: Hydrilla, Zostera, Vallisneria. All aquatic plants are not hydrophily, because you can observe, see that lotus, which is a hydrophilic plant, but that will never undergo for hydrophilic pollination. It undergo for insect pollination. In different agency it will carried out or takes place. So how we can observe, see that there are two types of pollination, or two. What happen in hypohydrophily and epihydrophily, that we will discuss here carefully. So in hypohydrophily, we can observe, see that pollination takes place below the surface of the water. In Zostera or in seagrass, we can observe, see that pollination takes place, and that plant is most probably monoecious. Monoecious means a single plant which produce male and female flowers, and in that pollination takes place below the water, and pollen grains were having without exine. So mucilage cavity is present or occurred, and pollen grains were actually exines absent in Zostera. Pollen grains are weightable, means here you can observe, see that they were having a particular weight, density. Because of that, they still remain below the water. And second type, that is known as epihydrophily, that indicates epi means above. So on the surface of the water, actually this pollination is carried out or takes place. Vallisneria is the example of epihydrophily, commonly known as eelgrass. So what happened in epihydrophily? Here two plants are there. It means dioecious condition takes place. One is male plant, another is female plant. So male plant produce male inflorescence which is below the water, but when male inflorescence will mature, that will release the male pollen grains or male flower at the top of the water level, and because of that, male flower when comes above the water level, it will undergo for anthesis, and female flower which is having a coiled or ribbon like structure, and when it matures, that ribbon or that is spring like structure, it actually uncoiled and comes above the water surface, where already male flowers were undergo for anthesis, and when female flower come, that pollen grain and pollination is easily carried out or takes place. These are the two types of hydrophily. In that hypohydrophily and epihydrophily, examples are most important. Zoostera are the example of hypohydrophily. Vallisneria is the example of epihydrophily. Commonly, these examples are known as Zoostera as a seagrass and Vallisneria is known as eelgrass. So next we will discuss about now biotic agencies, living agencies. Pollination takes place with the help of living agencies. So zoophily जिसको हम commonly नाम देते हैं, in zoophily how this pollination is carried out or takes place. Most probably all biotic agencies were showing some special floral adaptations, and these floral adaptations are common for Entamophilus, or we can observe the Ornithophilus or Chiroptophilus. Small changes are there that we will discuss laterwards, but common characters are there in biotic agencies. That characters are flowers are large, big, conspicuous in nature. It means visible in nature. Flower produce bright colored corolla, sometimes bright colored sepals also formation like in bougainvillea. So flowers produce scent, fragrance, and nectary glands were production. Pollen grain exine is spiny and sticky, that indicates pollen sac or pollen kit is present or occurred. So here we can observe, see different type of floral adaptations which takes place in insect pollination, ornithophilus pollination and chiroptophilus pollination. Let's go towards these pollinations. First is known as insect pollination, entamophily. So in entamophily we can observe, see that insect pollination, here is the example of insect pollination in salvia, which consists of special mechanism known as liver mechanism. So in first A diagrammatic representation, you can observe, see that divericate anther, diverent anther, which is having upper and lower lobe, where upper lobe is fertile and lower lobe is sterile. So when that insect enter into that flower to consume the nectar, it will first attach to lower anther lobe and indirectly upper anther lobe stick to that insect's their wings, their legs and their antennas. So you can observe, see that here this insect covered by lots of pollen grains, yellow dust like particle, that is nothing but a pollen grain, and that pollen grain travel from one flower to another flower and indirectly pollination is carried out or takes place. So honey bee is basically consist as a best pollinator in entamophilus pollination. This is second type, that is known as bird pollination, known as ornithophilus, or in bird pollination we can observe, see that hummingbird is most common bird, hummingbird is there, then sunbird is also there, crow, bulbul, parrot. In that also pollination is takes place with the help of these agents, and in examples are most important, like we know that bombax, agave, begnonia are the examples of ornithophily, and in chiroptophily, bat pollination. Here you can observe, see that at lower side where Anthocephalus, Adonsonia, Kigelia are the examples of chiroptophilus pollination. So when flowers are blooming at the night time and these bats were really come towards the collect the pollen grains which are edible, come towards the nectar, and because of that indirectly it come in contact with many flowers and pollination is carried out or takes place. So uppermost diagrammatic representation indicates ornithophily and lowermost diagrammatic representation indicates chiroptophily. After cross pollination, some questionnaires come to in your mind, that is how this cross pollination is takes place. that cross pollination is actually takes place because of few outbreeding devices. Because of few outbreeding devices, cross pollination is takes place. Instead of self pollination, cross pollination is takes place. And what are these agencies through that cross pollination is takes place? That agencies are, or devices are, declining. First is declining, that is also we can indicate or observe that formation of unisexual flower. So production and formation of unisexual flower. So we know that there are type of formation of flowers like staminate flower and pistillate flower. So only presence of single sex, that is known as unisexuality. So B, dichogamy. That dichogamy indicates that flower is bisexual, but that flower produce protoandry or protogyny. So protoandry means development of male gamete or formation of pollen grain first, then gynoecium. So protogyny means formation of gynoecium first, then pollen grain. So protoandry, salvia, rose, sunflower, in that we can observe, see that for protogyny, plantago, gloriosa, mirabilis, in that we can observe, see that protogyny takes place. So because of that condition, dichogamy, ultimately in that flower formation of only cross pollination. Second type we can observe, see that because of heterostyly, अगर heterostyly present है occurred है, it means when there is formation or pin or thumb head structure. So what is mean by pin or thumb head structure? So here you can observe, see that A means anther, or S means, here you can observe, see that stigma. A indicates anther, it means anther present at the lower side, stigma present at the upper side. So ultimately after anthesis all pollen grains come towards the lower side. So indirectly pollination will never carried out or takes place. Or in thromide, anthers are above and stigma is lower, so pollination is takes place. But what happen here, protoandry takes place, where first androecium mature, then gynoecium mature. Because of that here also cross pollination is only carried out or takes place. Third type you can observe, see that here self incompatibility. So what that indicates, self incompatibility, when pollen will not germinate on genetically similar individual. So this diagrammatic representation indicate that. So stigma, or you can observe, see that consists of S1 and S2 type of genotype. So all stigma consist of S1 and S2 type of genotypes. But that pollen grain which present at upper side, that is in first A diagram, S3 and S4, that will form a pollen tube. S3 and S1, from that only S3 will form a pollen tube. Why not S1? S1 is having stigma, or you can observe, see that female consists of S1 and S2 type of genotype. So S1, here no formation of pollen tube. Or in third diagrammatic representation, genotype of same plant S1, S2, S1, S2, because of that no pollen tube will form or occur. That indicates self incompatibility. So because of these circumstances or devices of cross pollination is carried out or takes place. Before we go towards the fertilization, we have to discuss about the pollen-pistil interaction. So here pollen-pistil interaction, all the events from the pollen deposition on the stigma until pollen tube enter to the ovule. So only a right type of pollen grain we can observe, see that will form a pollen tube. So right type of pollen grain ही pollen tube form करेगा, develop करेगा. और जैसे ही right type, ये देखो हम देख रहे हैं यहाँ पे एक pollen tube form कर रहा है, but that pollen tube travel through stigma, style and ovary. So because of obturator cells, because of chemical constituents, because of chemotropism, basically we can observe, see that their formation is carried out or takes place. Like chemicals, boron, inositol and sucrose level are important to formation of a pollen-pistil interaction. So a typical pollen grain consists of 3 germ pore, but from only single germ pore formation of a pollen tube, and other 2 germ pore undergo for germ furrow formation. So we know that these basic terminologies to formation of pollen pistil interaction. After pollen pistil interaction, our second event is carried out or takes place. That second event is known as fertilization. So fertilization means fusion of male and female gamete. But that fertilization is carried out or takes place after pollen pistil interaction, and various methods of entry of pollen tube into the ovule. So that pollen tube enter into ovule by micropyle, then it is known as porogamy. When that pollen tube enter by callosal end, then it is known as callosogamy. And when that pollen tube enter by integumentary region, it is known as mesogamy. So these are the 3 various types of entry of pollen tube into the ovule. Or in fertilization, when that pollen tube come towards the egg apparatus, where egg apparatus consists of 2 synergids and 1 egg cell. So first synergid cells consist of, here synergid cells consist of, firstly you should know that in synergid cells presence of filiform apparatus, and because of that filiform apparatus pollen tube come in contact with that, where pollen tube already consists of tube nuclei. Because of tube nuclei and filiform apparatus interaction takes place, hydrolytic enzymes were active and pollen tube burst. When pollen tube burst, tube nuclei degenerate and lower to tube nuclei presence of male gamete, sperm, that male gamete or sperm fuse to the egg cell and formation of a syngamy. So here syngamy takes place. First fertilization takes place, that is also known as formation of zygote, which laterwards formation into a embryo, where again second male gamete is there. That second male gamete come in contact with secondary nuclei and it shows double fertilization. So double fertilization is carried out or takes place in angiospermic plant. Or in angiospermic plant you can clearly observe, see that after fertilization what happened, after syngamy double fertilization is carried out. That indicates, or simply we can say that one of the male gamete fuse to the egg cell to form syngamy, and another male gamete fused to polar nuclei, central cell or secondary nucleus, is known as triple fusion or double fertilization, which basically carried out or takes place primary endosperm nucleus, which laterwards convert into a endosperm. Here a video which we can observe, see in details. So after fertilization, post fertilization structure and events. So what is post fertilization and structure and events? So after fertilization, whatever changes were carried out or takes place, that is known as post fertilization events. So in post fertilization events we will simply carry out external changes and internal changes. So external changes you can observe, see that where typical flower when convert into fruit, n number of changes is carried out or takes place, where sepals were fallen down. In few plant species we can observe, see that sepals were persist like brinjal, chili, tomato, but petals were fallen down, stamen fallen down, stigma style fallen down, but only ovary persist or remain. That ovary convert into fruit and ovary wall convert into a pericarp. That pericarp indicates fruit wall, which is again having 3 different layers like epicarp, mesocarp, endocarp, and inside that presence of ovule which convert into seed. So basically we can observe see that from external changes directly that plant comes to internal changes where internally outer integument of that is converted into testa, inner integument converted into tegmen, that funicle converted into stalk of the seed, and micropylate still present, that indicates micropylate. If nucellus present, that is known as presence of nucellus, and perisperm seed, or if most commonly it is consumed, perisperm seed present in Piper nigrum. Antipodal cells are degenerate, synergid cells are degenerate except in polyembryony, but egg cells are converted into zygote and that becomes embryo, and secondary nucleus convert into a pen or endosperm. So first we will discuss about the formation of embryo. That process is known as embryogenesis. So how embryogenesis is carried out or takes place. So that embryogenesis is actually carried out or takes place in different steps by different process. You can observe see that embryo will form. So embryo develop by different stages like pro-embryo, globular stage, heart shape, या फिर mature embryo को हम torpedo shape या फिर horseshoe shape भी बोल सकते हैं. So how it will develop or occur, that we will discuss. So that zygote, first zygote undergoes mitotic division, and when zygote undergoes mitotic division, it forms two-celled structure. From that two-celled, first cell is known as basal cell, which is present near to the micropylate, and another cell is known as apical cell or terminal cell. So latter word, that basal cell or lower cell is converted into suspensor, and apical or terminal cell converted into a embryo. So how their formation and development of suspensor and embryo simultaneously, that process is carried out or takes place, and formation of a suspensor and embryo. So one by one we can observe see that. So basal cell again undergo and formation of two-celled structure. From that two-cell structure it will again undergo and become four-cell structure. From that four-cell structure it will again undergo for division and becomes 8-cell structure. So with respect to the most commonly we can observe see that suspensor is produced in 6 to 10 cell structure. From that lowermost cell, which is large cell, which is present near to micropylate, known as hostorial cell, and the uppermost cell is known as a hypophysis cell. So that suspensor is basically formation and development of that embryo or lift towards the कैलेजल एंड, उसको कैलेजल एंड के पास ले जाने का काम सस्पेंसर सेल यहाँ पे कर रहा है. बट दैट एम्ब्रियो फॉर्मेशन takes place by terminal cell, where terminal cell undergoes, here you can observe see that two transverse division is takes place, or two TS will be takes place or occurred, or one LS will be occurred or takes place. So with respect to that, when we can observe see that there is formation and development of divisions, and through that divisions we can observe see that development of a globular embryo. So globular embryo formation we can observe see that here how it will form or occur. Through we can observe see that when two longitudinal sections are takes place and one transverse section is takes place, globular stage appear. That globular stage convert into heart shaped structure. That heart shaped structure converted into torpedo, or that indicates a horseshoe shaped structure. तो ये development कैसे होता है, form होता है by n number of mitotic divisions. We can observe see that their formation is taking place. So after development of embryo, or in dicot embryo or in monocot embryo, how that embryo basically form or occurred. In typical dicot embryo consists of embryonic axis and two cotyledons. Embryonic axis consists cotyledon, that is epicotyle, terminal part of epicotyle consists plumule. So we know that from plumule formation of shoot system, from radical formation of a root system. So basically we have discussed in that, but in dicot embryo we can observe see that. But in monocot embryo, monocot means only single cotyledon is there. Like in most of the grass or grass family, you can observe see that cotyledon is basically known as scutellum. So scutellum is here. Embryonic axis is there, but that embryonic axis consists second most part which actually differentiate that colorrhizae and coleptile, where colorrhizae is outermost covering of radical and coleptile is the outermost covering of the plumule. So that we can see in diagrammatic representation where that shows overall formation and development. But two types of seed basically present or occur. One is known as albuminous seeds, presence of endosperm, and another is known as non-albuminous seeds where absence of endosperm. So when endosperm present, it is known as albuminous, and when endosperm absent, it is known as एक्स-albuminous. तो albuminous और एक्स-albuminous ये types भी हम यहाँ पे observe कर सकते हैं, देख सकते हैं. So these are the diagrammatic representation of seeds. So dicot seed or a monocot seed, you can clearly observe see that how their differentiation is there, how their formation is there. In dicot seed, endosperm is absent because that is consumed, or in monocot we can observe see that endosperm is present. So not in all dicot it is basically consumed, but in most of the dicot plants we can observe see that this is bean shape, so it is absence of endosperm. So now we will discuss about the new terminologies in flowering plant, that is known as apomixis. Apomixis, away from mixing, and polyembryony means without mixing of gametes, formation and development of new individual. So apomixis is studied by Winkler in 1908. So it always show that it mimics the sexual reproduction. It means it actually looks like the sexual reproduction, but that is not a sexual reproduction. Why it looks like the sexual reproduction? Because it produces seed. Seed production is there, and seed production is done by the apomictic formation, that is known as apomictic seeds. So genetically similar individuals were produced by apomixis, so it is known as asexual method. So gametes are produced, clones are produced. Opuntia, onion are the most common type of apomixis. Polyembryony studied by Rao in 1965, where many embryos were produced. So many embryo production is done by simple polyembryony, where fertilization is more than one egg cell. So more than one egg cell fertilization takes place, where many egg cells are present and fertilization takes place. So citrus is the common example of simple polyembryony. Cleavage polyembryony, where you can observe see that proembryo split into many parts, like in twin, monozygotic twin, a single zygote divided into two, like this. Proembryo split into many parts, and because of that formation of many embryos, like orchid, pinus, in that we can observe see that. And third type, that is known as adventative polyembryony, where formation of embryo from other than egg cell. So other than egg cell, it means that may be antipodal cell, that may be synergid cell, that may be nucellus, that may be integuments. So onion, trillium, in groundnut we can observe see that adventative polyembryony. And there is another type that we will discuss in another topic, that is known as parthenogenesis. I hope you like that video. So for that, thank you.